sadly died when he was 21, and his sister Chorea also died of juvenile Huntington's disease, as well as his father at the age of 33. So those of you who work in Huntington's disease know that it's a disease of families as well as individuals. And this is an old video from the Movement Disorder Society, but I think it's an important video because this is what ballistic chorea looks like in Huntington's, and it's incredibly difficult to treat. And actually, we don't have good treatments for ballistic chorea. And as you can see, it's very, very disabling. And as the disease advances, in advanced stage disease, the movements disappear. Patients become akinetic, rigid, mute, unable to speak, unable to swallow, and this period of time can last many years. So people in families often know what's coming while they themselves are at 50-50 risk of the disease. And voluntary euthanasia is a really big subject in this field. And in Canada, where medical assistance in dying is legal, there are many people with early Huntington's who've signed up for it. So it's indicative of the huge burden this disease places on both individuals and families. So this is from a recent review with potential therapies that are in development, targeting the DNA. Obviously, CRISPR therapies are in development, are very challenging with delivery. Zinc finger proteins that are transcriptional repressors for Huntington are also in development. We're doing some work on this in the lab. Big interest in DNA repair modulators, modulating the CAG, small molecules affecting splicing, ASOs, and RNAi. So I'm going to talk in two parts. I'm going to talk about the challenges of therapeutic development and talk to you about the tominersen story, and then also the opportunities in new molecular mechanisms. And I'm going to focus on DNA repair, gene therapy, and allele selective targeting. And so Huntington lowering. So there are over 50 published papers in mice where you switch off mutant Huntington expression, whether at the DNA level, which is by far the most effective, or with ASOs or siRNAs, you can reverse symptoms of pathogenesis in the mice. So Huntington lowering in mice works. But we have a big challenge in translating therapies from mice to humans, and that is indicated here. This is a mouse brain. This is a rat brain, and this is a large rhesus monkey brain, and this is our brain. And really, the challenge in therapeutics is getting therapies to the whole human brain. And in Huntington's disease, we want to treat the cortex, and we want to treat the deep structure of the striatum. So we can actually pretty easily cure a mouse of Huntington's disease, but humans is a huge, big challenge in comparison. A mouse brain is literally tiny. So ASO therapy, antisense oligonucleotides are single strands of chemically modified DNA. They're designed to bind to the mRNA of Huntington. And the result is that you get a DNA RNA hybrid, and you get degradation through the RNase H1 pathway, and you get less protein being made. So the preclinical data for tominersen showed sustained Huntington lowering in non-human primates. There was a 50% reduction in Huntington in the cortex post-intrathecal dosing. And you can see here significant lowering in the cortex, 50% roughly in the caudate and the thalamus, which are deep structures. And the one thing about ASOs is that they last a long time. They stay within the cell, and in this study had an 8-week duration of action. And the toxicology program in non-human primates identified a very wide safety margin. There was no preclinical toxicology at doses that were 7 times that used in the first in-human studies. So there was a lot of excitement. We developed a fast push intrathecal bolus technique that we hoped would get the ASO to the brain. We took off 20 mils of CSF. We gave 20 mils of drug in artificial CSF over 3 minutes. And then we asked subjects to walk fast for 30 minutes to increase CSF flow. And everyone was very excited when in December 2017, when the trial read out, that for the first time with an intrathecally delivered ASO in a toxic gain of function disease, there was successful lowering in the brain of a protein. And this was the dose-dependent lowering of CSF mutant Huntington from placebo right through to the 120 milligram dose. So there was a lot of excitement with this, and it kind of made headlines throughout the world because everyone thought that a treatment was going to be around the corner. And the program was taken over by Roche, who licensed the ASO from Ionis, and Roche evaluated it in an open label extension, and data from the open label extension informed a phase 3 study. Now, they were very keen to go to a phase 3 study because this was a rare disease, huge unmet need, and they had some data from the open label extension. So they went to a phase 3 study, which was over 2 years, comparing tominersen 120 milligram dose every 8 weeks. You'll note that's the top dose that was used in the phase 1. Tominersen 120 milligrams every 4 months, 16 weeks versus placebo, 791 subjects, all symptomatic disease. This was a 2-year study. And then on the 22nd of March, 2021, the Data Safety Monitoring Board recommended that the study dosing be halted and stopped because the 8-weekly group were doing worse than placebo on the unblinded data they saw, and it was recommended to stop the study. And this is the total functional capacity, which is a functional score. This is baseline. This is week 69 when the study was stopped. This is 8-weekly, 16-weekly, and placebo. And you can see here the 8-weekly group is doing clearly worse than the placebo at week 69, which was month 17. This paper, I've said revision, but actually this paper was published on 7th of December in 2023. And so all of this data is available both in the paper and a huge supplementary section. What was important as well is that it was noted that the ventricles were expanding, but there was no associated increase in brain atrophy. So this is the 8-weekly group, orange is the 16-weekly group, and this is placebo, whole brain, caudate volumes. But you can see here the ventricles there was a dose-dependent increase in ventricular volume, and it's very clearly dose-dependent, placebo, 16-week, 8-week. And this is CSF neurofilament, CSF protein, CSF tau. This is the original phase 1 study where NFL was measured. And in this study, some of the subjects at the 120 milligram dose were beginning to get increases in CSF NFL. So it was there in the original study. This was the open label extension. There was a 53% mean increase in NFL at 15 months in the 4-weekly dosed group. And in the 8-weekly dose group, there was a 10% increase in NFL. So there was a spike and then an increase that stayed slightly up. This is time after first dose and percentage change from baseline. This is the lowest exposure to the highest exposure. So the highest exposure had this big spike in CSF NFL, and the ventricles got bigger. There was very spectacular mutant Huntington lowering, and there's an increase in CSF protein. And you can see here going down to placebo, there is some increase in ventricles because that's an age-related effect. There is an age-related increase in CSF neurofilament, but no increase in protein, and obviously no Huntington lowering. The lowest exposure there was some increase in ventricles, but it wasn't significant different from placebo. A very subtle increase in NFL and lowering of Huntington, and no increase in CSF protein. And NFL went back down. So, don't know what happened there. So, these data, I think, were very informative. And so what went wrong? So exposure, the 120 milligram dose was chosen. And because with ASOs that are delivered intrathecally, to get to the deep structures of the brain, like the striatum, you have to have a relatively high dose. So the 120 milligram dose was chosen was because they wanted to maximize striatal penetration with an intrathecal delivery. And the dose was too high. And I think now CSF neurofilament going up is now recognized as a safety biomarker, and it's recognized by the regulatory agents as well as a safety for safety. The high dose and high exposure was appearing to cause chemical inflammatory effects with high CSF protein and white cells, and resulting, my hypothesis and our hypothesis, impaired CSF resorption dynamics, possible CSF outflow obstruction, and you get enlarged ventricles. And that's actually been seen with some other ASOs. It was a non-allele selective approach, so we cannot dissect ASO toxicity effects at the high dose and lowering maybe too much wild type Huntington. So that's an important point. We can't dissect it. There was lack of efficacy. It was a late symptomatic patient population. There were subjects with total functional capacity of TFC 5 and 6 with CAP scores of 900. So there were really some very late stage patients. The 16-weekly cohort was stopped early at 17 months instead of 25 months. And it might be that they weren't doing worse, but 17 months, we know in studies, is probably not long enough to see a difference. So we don't know what would have happened if this was continued. Exon 1 was not targeted directly, and that may play a role. And so I think it's a combination of effects. The dose was too high. It was late stage patients, and it's difficult to dissect wild type Huntington effects and the ASO toxicity. And I think we just don't know the answer to that. I do think it may be, my personal thing is it's a combination of both. Importantly, none of this was predicted from the 15-month preclinical non-human primate data, which didn't show any of this at very high doses. But I think the important point here is when we do, and we all do GLP tox, it's in non-diseased animals, and then you're giving these sorts of drugs, which can be pro-inflammatory, into diseased brains. There was a hypothesis-driven post-hoc analysis of Generation HD1. It was really, is there any data to support continuing the program? And are there any people who were younger, earlier stage in disease with lower drug exposure who may have any benefit? And this is a post-hoc analysis, so it needs to be tested. But there was some suggestion that those who were under the age of 48, who were earlier in disease, may have some potential improvements on the clinical scores. But it's a post-hoc analysis. It needs to be tested. Interestingly, in the 16-weekly group, the plasma neurofilament showed trends below placebo beyond week 21. And so this is the placebo is the dotted line, and you can see here the 16-week group was clearly below the dotted line and continued below at day 101. So week 101. So there, you know, this is interesting, and it generates new hypothesis potentially. And so what's important is in some subjects who were in a natural history study, they rolled over into getting the drug at 16 weeks with 120 milligrams, but they were not given a loading dose. In the original phase 3 trial, all of the subjects were given a 120 milligram loading dose 4 weeks apart, because the modeling of pharmacology that Roche did, it was very much wanting to see if they could get to steady state with Huntington lowering. But here you can see, this is NFL, again, 120 milligrams every 4 months. There was no loading, and then there was no increase in NFL. So it does suggest that that spike that was being seen was the acute loading of 240 milligrams over an 8-week period. And I think the data is supportive that NFL increases can be avoided at lower exposure. So this has generated a new phase 2 study, which is a dose finding study and a safety study to determine if there's a safer lower dose in younger, earlier patients with no loading. And this study is going on worldwide. It's about 50% recruited, and it's a dose finding biomarker study. So we wait and see. I think also that importantly, we do have to be developing allele selective approaches. This is very challenging because to get a true allele selective approach, currently you can either target the CAG tract, which has challenges of off-target effects because there's lots of mRNAs with CAGs in them, or you can try and target a SNP that is then linked to the mutant allele, because there's no difference otherwise between the normal and the mutant apart from the length of the CAG. So there is an allele selective approach in development with 2 different ASOs, but one from Wave, one from Roche, targeting mutant Huntington only. It is personalized genomic medicine. It can only target those carrying the SNPs. So it's the current SNP that will be tested is about 40% of the population of Western European origin. And it is more challenging because you have less of the mRNA real estate. And so developing potent molecules has been more challenging. But that's in development. It needs a phasing assay. So people would get genotype to see if they have the SNP, and then you have to do a long read sequencing phasing assay to see if the SNP is on the mutant allele and not the wild type, because you don't want to lower the wild type and preserve the mutant. So the phasing assay is very important. So I think now we're at this point where I think we need the next generation of RNA drugs. We need better chemistry, improved backbone chemistry, stereochemistry, conjugation chemistry, delivery with bioconjugates and nanotechnology. And we need to be able to have nucleic acids that can deliver to the whole brain. That's our big challenge. And so the top dose of tominersen was chosen because they wanted to get to the deep structures, and we need to treat the striatum. And so with an intrathecal delivery, it may be that we have to also think of different delivery ways. Maybe we should be delivering ICV for the striatum. I'm part of a big consortium that has been given £8 million over 3 years to address these challenges. It's academics and industry really trying to see if we can get better linker chemistries, nucleic acid cellular uptake, endosomal trafficking, PKPD in CNS, heart, and skeletal muscle. So I think this is really important. It's got many people worldwide. And so another program that I think is interesting is a conjugated siRNA from Alnylam. This is moving towards the clinic. We're gearing up towards a first-in-human study. This is a single dose of a Huntington siRNA, and they have data in non-human primates showing that there is very good lowering in the dorsolateral prefrontal cortex, but also in the caudate, and that's after 3 months after an intrathecal dose. So with siRNAs, you can actually, they tend to be more potent. They still have the issues of nucleic acid therapeutics, but I think it looks like from studies in APP and Alzheimer's that the siRNA can last up to 6 months with a single dose. So genetic therapies. zinc finger transcriptional repressors I'm going to briefly talk about, small molecules targeting DNA repair, and the AAV gene therapy. So there is a AAV gene therapy program that's currently ongoing. It's in the UK and Poland and Germany and the US. It's the Unicure microRNA Huntington. We're involved in the study as well. This is an AAV5 microRNA against exon 1 total Huntington. And it is intraparenchymal surgery to the caudate and putamen. And the surgery here is intrastriatal convection enhanced delivery. This is very much speeded up. The surgery takes between 12 and 15 hours, but this is real speeded up data from a neurosurgical delivery from the trial. The data so far, this is the data that was read out in June last year. This is the AMT low dose in yellow, the AMT high dose in red, and this is natural history comparator data. It does suggest that there is beginning to be some separation of the total motor score, the symbol digit, potentially the composite UHDRS, and potentially the TFC from historical controls. The study is ongoing. The 30-month data was read out recently as well. These are very small numbers of people, but the clinical measures suggest they're going in the right direction. But again, this is small numbers of people, and the historical control group is needed because the sham in this study, which is in purple, was done for 1 year. So I'm going to move on to an area that's of super excitement in repeat expansions diseases, and that's DNA repair and repeat instability. So there are over 50 known repeat expansion diseases, Fragile X, Friedreich's ataxia, Huntington's, spinocerebellar ataxias, myotonic dystrophy, and there are a number of the genetic modifiers that I'm going to talk to you about also affect these other diseases. And so in Huntington's disease, we know that somatic expansion of the repeat drives disease progression. And we've known for a long time that there's a huge residual in the genetic variation and age of onset. And so this is someone with 42 repeats. And you can see here, that this is 42 repeats. This is from a large amount of data. And you can see, look at the difference in the age range. So between 40 and 50 repeats, there's a lot of spread in the age range. And what we know is the repeat expands throughout lifetime, particularly in the brain in postmitotic neurons. So you're born with 42 repeats, but that we know that expands in the brain. And this is from an old paper from Peggy Shelbourne. This is small pool PCR in the striatum and cortex. And look at all the long CAGs she detected. Some CAGs are up to 700 in the striatum. And we know from many people's work, Vanessa Wheeler's work, Darren Monkton's work that we did in collaboration with him, that somatic expansion in brain or blood, the rate, and blood is just a proxy for the brain, modifies age of onset and disease progression. And Vanessa very nicely showed in postmortem brain that patients with more somatic expansion had earlier HD onset and patients with less expansion had later onset. And Davina Hensman-Moss in my lab, when she was a PhD student, did a GWAS of the track data. And actually, she found a significant GWAS hit in MSH3. And this was before this came up in the big GEM consortium. And this was interesting because it had been suggested to be a modifier in mice, but not in humans. And so what was interesting is this was a beautiful example of reverse translation, how something that had been of big interest in mice, modifying somatic expansion, was then shown to be a powerful modifier of progression in humans. And we went on and looked at this in more detail. Mike Flower in the lab showed that it was modified both myotonic dystrophy and HD, and it was a result of reduced expression of MSH3. And what we think is happening at the repeat, and this is a very simplistic model, but during Huntington transcription, the structure of the expanded repeat is such that during the unwinding, there is loop outs, small CAG loop outs that occur during repeat expansion. And what happens with those loop outs is they get recognized by MSH3 and MSH2, which is mutase beta, and then MLH1 and PMS2 come and they nick the DNA, and then XO1 and POLG, so XO1 cuts out the DNA, and you can see it's on the opposite strand, and then POLG and LIG1 fill in the gap. But what happens, as you can see, is when the gap is filled in, the CAG is expanding. Now, this is what we think is happening. There's a lot of data to support this model. And the players involved in this have all been look like they're important in the GWAS. So what we know is that MSH3 promotes the Huntington repeat. If you knock out MSH3, you don't get that expansion, because MSH3 recognizes the loop out. And we know that somatic expansion promotes production of toxic proteins and exon 1. And we've done a lot of work with an MSH3 ASO. And we found in HD-derived medium spiny neurons from iPSCs that the MSH3 ASO completely ablates somatic expansion in these neurons. And so MSH3 is a huge area that is in people looking at this for drug development and clinical trials. And so what we think is happening in Huntington's disease, and I think this is probably similar for other repeat expansion diseases, is that a rate driver is the rate of somatic CAG expansion in post-mitotic neurons in the brain, in brain diseases. It depends on the CAG length, and it's modified by certain DNA repair genes. And that expanded CAG then results in a toxicity driver that is triggered when somatic expansion reaches a threshold CAG length. Now, some people think it's 90, some people think it's 150 from data that both Nat Hines, who's published, and Steve McCarroll from the Harvard has presented. And that results in more production of the alternatively spliced toxic exon 1 and expanded polyQ protein and fragments. So this is dependent on intact MSH3, and MSH3 is a huge target. This is data that we generated in the lab in iPSC-derived medium spiny neurons. If you knock out the ATPase of the MSH3 here, and you ablate the ATPase, it behaves like an MSH3 knockout. And you ablate somatic expansion. And so I'm very involved in drug discovery program in this, but me and many others are working on MSH3 targeting molecules and approaches for somatic expansion and to stop somatic expansion. And then I'll just finish and tell you about the zinc finger protein, which is an allele-selective gene therapy program in development. And this has been in development a long time, but it's now back on for development. And the reason I think this is really important is it targets DNA. And if you target DNA, you stop somatic expansion because this is targeting Huntington transcription. So it targets everything. It targets the toxic protein, it targets the repeat, because if you do not have Huntington transcription, you do not get somatic expansion, because it's somatic expansion is dependent on transcription through the Huntington gene. So the ZFP has been engineered to bind to longer repeats, and it has a functional domain, which is the actually the CRAB protein, which is a transcriptional repressor that switches off transcription. So the Huntington is not, transcribed. So you get no Huntington mRNA, and you get no Huntington protein, and you get no somatic expansion. And this is from a paper a few years ago from Sangamo, and with CHDI, who showed that striatal delivery of the ZFP in knock-in mice, was mutant Huntington specific. It cleared aggregates, but it also reversed some of the PD10A functional loss of PET in the striatum. So this went forward for clinical development, it stalled, and now it's back on for clinical development. And the goal for this is a single dose administration, targets Huntington transcription, and there's a lot of work now to get the ZFP transcriptional repressor towards GLP tox studies. And so this is an overview of therapeutics in clinical and preclinical development for HD. There are many splicing modulators. PTC is in the clinic, many RNA modulators. Annexon has an anti-C1q antibody in trials. A number of oligonucleotides. I've talked about Ionis, Wave, Vico. Alnylam is going to be starting this year with the conjugated siRNA. Unicure is in gene therapy trials, but these other programs are following behind. Many approaches trying to develop drugs against the repeat expansion, and these are some of the companies, and protein clearance. So it's a busy space, and but it's a difficult disease. So I think anything that's found in HD will obviously have implications for all of the other repeat expansion diseases, particularly those that are targeting the repeats. And cell replacement therapy is also of interest with transplantation for replacing lost neurons. And so one of the things in Huntington's disease and many other rare genetic diseases is that we have a genetic test. And so a lot of my research has also been focused on when is the best time to treat. And we know from our Huntington's young adult study that was published in 2020 is that young adult HD gene carriers 24 years before their predicted genetic onset have normal brains, have normal CSF neurofilament, and there is nothing to find. So ideally, we want to treat then to try and prevent this disease. And as part of that, I led a consortium to develop a new Huntington's disease integrated staging system, which was published a year and a half ago now in the Lancet Neurology, which was basically a staging system of Huntington's from birth to death, with a stage 0 to stage 3, and a bit like the cancer staging system, 4 stages. And stage 0 is this point 24 years before onset, when there's nothing to find and there's just the presence of the gene. And so I think in the future, we want to use the staging system, and we want to use the ongoing work that we're all doing to be able to treat then at stage 0 and stage 1. And I genuinely think that that will be our greatest chance of actually preventing this disease ever occurring. And that's really what we all hope. And I think my vision as someone who's worked in Huntington's disease for a long time is a world in which it's not something that families have to worry about. And that's very important. And I think finally, it's obviously going to be a big collaboration, because the world is basically one big laboratory, and it's by us all working together with clinicians, families, and scientists that we will achieve success. And success is going to be incremental. Success is not going to be like SMA and nusinersen. For neurodegenerative brain conditions, we have many more challenges. So I'd like to thank you for listening. I'd like to thank all of the people in my research team. I don't know whether this is my lab team, my clinical trials teams, my HD young adult team, the HD Centre, and I'd also like to particularly all the patients and families who contributed to my research over the years. And thank you for listening.